Welcome. Today we're going to start with chapters 5 and 6, which pertain to lecture number 2 of your microbiology lecture. What is metabolism? Metabolism means basically what the different biochemical reactions that organisms do in order to either break down energy or to make the energy they need in order to survive. Some of them include simple ones such as fermentation, and others include very complex ones such as cellular respiration or lipid metabolisms. We're also going to go over a little bit of your microbial growth and nutrition. And what I mean by know your trophs is a Greek word which means food. How do they obtain the food? Please see the video in your study area. So here we go again. Metabolism is the collection of controlled biochemical reactions that take place with the microbe. And what we mean by that is how they actually generate energy or how they produce energy. Catabolism versus anabolism. When we talk about catabolism, it refers to how organisms break down larger nutrients or anything that is large, such as biochemical reactions, in order to make smaller ones, and in the process they make energy. Okay, the opposite will be anabolism. One thing that we have to understand, when we break down these micronutrients, it is not a one-step process. It is a very series of steps in which many enzymes are involved. So therefore, it is not one spontaneous reaction. It is a series of many reactions which at the end will give you the energy as a form of ATP. As you can see here, you can see what is the catabolism, which breaks down, again, molecules. You make energy. However, as you know from the second law of thermodynamics, energy is not fully gained. You always lose some as heat. But what is the purpose of making this energy? This energy will be used for anabolic reactions. In other words, to make larger building blocks known as macromolecules, which are needed for cellular processes such as cell growth, cell division, or cellular structures that are needed by other organelles in the cell. Let's look into the different catabolic pathways. The main ones that we're discussing here are your carbohydrate metabolisms, lipid metabolisms, and your protein amino acid metabolism. In the carbohydrate metabolism, we refer to the ones that start with glycolysis, and they might go through fermentation or cellular respiration. Then you also have the pentose phosphate pathway. We will produce ribose and NADPH from your glucose 6-phosphate. Then you also have the ones that are known as your glycogenolysis, which break down the glycogen to eventually glucose, and from glucose you will move on to glycolysis. In lipid metabolism, you have the beta oxidation pathways. And basically what happens here with the lipase enzymes, the fatty acids are removed from the glycerol, and then they are slowly broken down into two carbon molecules known as acetyl, and acetyl-CoA will be formed in order to join later on into cellular respiration. You also have the ketone bodies, which are basically broken down and used for energy by muscle cells and neurons. Your protein amino acid metabolisms refers as forming amino acid pools. These free amino acids will also be used later on to build other protein necessary for the cell to be maintained. There's a process known as transamination, in which your amino end is removed. And your oxidative deamination is the amino group the glutamic acid is removed, and then it forms alpha keto acids. These also will form part of the urea cycle, which produces the urea from the ammonia, since our cells or any microbe cell cannot really process for a benefit. In the case of anabolisms, we have examples such as photosynthesis. Photosynthesis, which you know, it happens with organisms that happen to have the organelle chloroplasts. We have glucogenesis, glycogenesis. We also have the anabolic formation of fatty acids from your ketone bodies. You also have the formation of lipids. It is the reversal breaking down. Remember, this is anabolism. So you join your fatty acid with your glycogen, with your glycerol, excuse me. Your complex lipids are also formed here, with a series of enzymes known as kinases. The amino acid synthesis is also necessary. As you may remember, we have 8 essential amino acids that, in the case of eukaryotes, are not produced, and they get this from nutrition. Okay, nucleic acids are also necessary to form new bands of DNA formation and RNA formation, and your proteins and peptide hormones are used to make from the amino acids to proteins in order to be able to do a genetic expression during messenger RNA and transfer RNA during the translation process. What are the basic chemical reactions underlying metabolism? Well, these are known as redox reactions. An oxidation occurs when basically a bond is broken and the electrons are given away. And when the electrons are given away, they are actually picked up by coenzymes, and then they are again given away, and the final acceptor will be known as the reducer. So when you get reduced, basically that means that you are actually gaining those electrons, and 3 important of the electron carriers that I'm referring to could be NAD+, NADP+, or FAD. NAD+ and FAD happen to be part of your cellular respiration processes. NADP+ happens to be part of your photosynthetic process. And this diagram basically informs you what I mean by redox reaction. As you can see, the electron donor gives it away, and the one that gets it is known as the electron acceptor. So this electron acceptor just got reduced, and when it gives it away again, it becomes oxidized, and then that's when that receives it becomes reduced. See video in your study area in mastering. The basic chemical reactions underlying metabolism. Your cells, in order to survive, or any microbial cell in order to survive, needs energy. And the final form of energy is known as adenosine triphosphate, which is ATP. When we actually hear of ATP, it means it has 3 phosphates. But before it has 3 phosphates, it has 2, which is ADP. And by getting the third one, it just got phosphorylated. The way you make ATP in the cells, it's either through substrate level phosphorylation, oxidative phosphorylation, or through photophosphorylation. We're going to see these processes next. Anabolic pathway uses some energy of ATP for breaking phosphate bonds, so therefore they need a little bit more energy in order to go through these processes. See this video in your study area of mastering about the enzymes. Here you have some enzymes, examples which are involved in these catabolic and anabolic reactions. As you can see, an enzyme always ends with the 3 letters ASE. The enzymes are mostly named according to their function. For instance, if you see the ligase, it means to ligate, means to put together or joining. Okay. Polymerase means to make polymers, and so on and so forth. Enzymes are proteins. However, the roles in the enzymes is to actually be activators or inhibitors. inhibitors. Inhibitors, When we look at the enzymes active site, sometimes it has also inorganic cofactors and coenzymes, which are organic cofactors. These are necessary for some of the enzymes, not all of them, to actually become active when the substrate binds to the active site. Once the substrate binds to the active site, this is when the enzyme starts doing its work. This table indicates some example of those cofactors. And as you can see, some of them happen to be either metal ions or coenzymes. In order for the process to occur in the enzyme, the active site has to be open and the substrate has to be correct. In other words, what it means is that enzymes are very specific to the substrate. What's important is that the energy is reduced by the enzyme, the activation energy that is, so therefore it makes this reaction happen spontaneously, okay? Without the enzyme with the active site open, it will not occur. And if the active site is not correctly shaped for the substrate, then the activation energy would not be there and it would not occur either, because you can see it would take much energy to start it, which it would not give a correct or possible yield. As you can see, the active site has to be very similar to the substrate shape. If not, it would not occur. In this example, you can see in label number 1, the enzyme here happens to be fructose 1,6-bisphosphate aldolase. That means that it is the enzyme that can only accept the substrate fructose 1,6-bisphosphate. Once it binds to it, the enzyme will form something known as the enzyme substrate complex, and in this case, this enzyme happened to be a catalase, meaning it's going to break down the molecule, and then the 6-carbon molecule is going to become 2 3-carbon molecules such as G3P. See video in study area. The roles of enzymes and metabolism depends on many factors such as temperature, pH, how much enzyme versus substrate concentration there is, and the presence of inhibitors which might block the enzyme's active site. Now, these inhibitors do not really denature the enzyme, they just happen to stop its function. For instance, most enzymes in your body happen to work best at 37 degrees Celsius, which is the standard temperature that we survive the best in our case, and that is also for most of the bacteria. As far as pH, again, same thing. The best pH is like water, around 7. Now, when we talk about substrate versus enzyme activity, you will have so much substrate in which the enzyme can break down, but once it reaches the saturation point, meaning that there's so much more substrate than enzyme, it can only go and do so much and not go any further. Denaturation of protein enzymes occur when they're not in the correct environment. For example, when we talked about temperature or pH. A functional protein must have the correct shape. If it becomes denatured, it becomes unwinded and therefore loses its shape and function. Enzymes can go through competitive inhibition or non-competitive inhibition. In the case of competitive inhibition is when you have your correct substrate and you have a competitive inhibitor that is, like they call it, competitive. It is actually competing for the actual active site. An example of that would be, for example, when you give patients drugs. Some of these drugs can be inhibiting the signal of pain. And then what's going to happen with that is that eventually your body, or in this case human cells we're talking about, will start producing more and more enzymes. So then you have to increase the competitive inhibitor, which in this case will be the painkiller, in order to sustain the pain for the patient. When you have a reversible competitor, what happens is it is removed, and then your substrate, correct substrate, starts binding to it. The allosteric control of enzyme activity, it's normal in your cell, and it is frequently used when you have enzymes that break down enough of a product or make enough of a product, and once you have reached that saturation point, another cofactor will bind to this area known as the allosteric site, and this allosteric inhibitor will make the active site, as you can see right here, change. And when it changes, it will not, the substrate, be possible to bind to it. Allosteric activation, though, is once you start with another feedback from signal, and the allosteric site is still going to be open, and the allosteric activator will go in, and it will make the actual correct shape of the active site so your substrate can bind to it. Feedback inhibitors are an example of how allosteric site functions. In this case, you can see when your pathway is operating, it makes your actual products from enzyme 1 through enzyme 2 and enzyme 3. And once you have enough of that product made, the end product made, the very last one will act as a feedback inhibitor, as you can see from here, and it will bind to the allosteric site, therefore changing the normal shape of enzyme 1 on the active site, and it will not allow more substrate to be broken down in this case. Would that change? Yes. As you recall, the actual allosteric site also can act as an activator to start making more product. Tell me why. How can oxidation take place in an anaerobic environment that is without oxygen? We'll talk about it in class. Carbohydrate catabolism. This means we're breaking down sugar. Many organisms oxidize carbohydrate as primary energy source of anabolic reactions. Okay, so you might say, what does that mean? Well, in order for you to make molecules, you need first the energy. So first thing to do is break down a molecule to get the ATP, and the ATP then is going to be used to make more new substance needed for the cell. First example here is glucose, and it is broken down by two processes, which is cellular respiration or fermentation, and that depends on if there is or not oxygen present in the environment. This is an overview of your catabolic process. Okay, as you can see, respiration goes through many more steps, and it is with glycolysis, and it moves on to the intermediate step, which is followed by the citric acid cycle, which is the same thing as the Krebs cycle, and through the electron transport chain in which oxidative phosphorylation occurs. In the case of fermentation, it is a smaller process, but at the same time, that's why you make much less ATP. In fermentation, you only make 3 ATPs at the end, versus 38, which is an average, by the way, it depends on the organism, in the cellular respiration process. This is the detail of the glucose catabolism. It starts with glycolysis. Glycolysis. Glycolysis is the first step of cellular respiration or fermentation. It starts with glucose, and the first phase, which is glucose, all the way to glyceraldehyde 3-phosphate, is known as the investment phase. And the reason it is called that way is because, as you can see here, 2 ATPs are used in order to break down glucose into 2 3-carbon molecules. After that, we call it the energy harvesting stage, in which we make the ATPs that you see right here. So the net here is not really 4 because, as you see, we invested 2. In the presence of oxygen, it will move on to the citric acid cycle, which is also known as your Krebs cycle. However, if there's no oxygen, it will go on to fermentation. Where does the glycolysis begin? It occurs in the cytoplasm of eukaryotic cells, and in the case of prokaryotes, which they don't have a mitochondria, the second component will not happen in the mitochondria, it will happen in the actual cell wall. The way it makes energy is through a process known as substrate level phosphorylation. This is the overall reaction. You start with glucose and you end up with pyruvate, which is known as pyruvic acid. See the video in your study area. Here's more detail. We begin with glucose in glycolysis, 6 carbons. It's broken down with the investment of 2 ATPs into 2 G3Ps. Then these 2 G3Ps are going to be oxidized, in which your coenzyme NADH gets reduced. These NADHs, in the presence of oxygen, will move on to the electron transport chain, in which they will also get oxidized for the use of making, at the end, ATP. However, we also have here 2 substrate level phosphorizations and 2 more here. Here is the phospho level phosphorylation. The substrate level phosphorylation, as you can see, involves the holoenzyme, and it picks up one of the products. Now you might say, where does this product come from? If you go back to the previous page, it's right here. G3P or PEP, which in this case, they're showing you PEP, which is phosphoenolpyruvate. Phosphoenolpyruvate has a phosphate, which is removed, and ADP is phosphorylated into ATP. Cellular respiration. As you may recall, at the end of glycolysis, we end up with pyruvic, which is also known as pyruvic acid. Pyruvic acid is going to go through oxidation reactions, and then it's going to get to form ATP by another series of redox reactions. It has 3 main stages, which is the first one is also known as the intermediate step. You will move on to the citric acid cycle, and here you will generate ATP and more NADH and FADH, which will go on to a final series of redox reaction in the electron transport chain, which is also known as oxidative phosphorylation. And here is the overview. Intermediate step, coenzyme A comes in, forms acetyl-CoA, and then your acetyl-CoA will bring in your 2-carbon molecule into the Krebs cycle. Krebs cycle is named after Hans Krebs, who is the one that actually was able to depict it in the 1930s. Here, much of the energy is going to be transferred to the coenzymes NAD plus to make NADH and FAD to make FADH. These are the main 6 types of the reactions. Which are here. So after the intermediate step, acetyl-CoA comes in with 2 carbons. As you can see, the two carbons here, they will join this 4-carbon molecule known as acetic acid and form the citric acid. That's why it's called the citric acid cycle. Then it's going to be summarized into the isoacetic cycle. And here is when this redox reactions start. Once the isocytric cycle becomes right here oxidized, NAD plus becomes reduced, and it's going to go to the electron transport chain. And you can see here the carbon dioxide is also formed. Now, as you may recall from your readings, the main formula for the cellular respiration is glucose plus oxygen gives away carbon dioxide right here, and water, and of course indirectly ATP. Now this is not the first time you see the carbon dioxide. You also saw in the intermediate step previously, and here again is another carbon dioxide. So from the formula, you know you have 6 carbon dioxide produced. Well, here you have 1, 2, and in the intermediate step here, right here, you had another 1, but it's really 2 because there's 2 pyruvic acids coming in. So that's where you get those 6 carbon dioxides formed. Now what happens to the alpha ketoglutamate acid is going to become oxidized, and NAD plus becomes reduced. And you form your succinyl-CoA. Now here you have GTP, which is going to release its phosphate to more ATP. And the way this happens is because the coenzyme A that came in is going to release its bond in order to get oxidized, and this redox reactions here you see again. So technically here you don't see in the picture what's happening is that ATP is formed through substrate level phosphorylation. Here is the new coenzyme that you haven't seen yet. FAD is becoming FADH2. Now the question is, what's the difference between FADH2 versus NADH? Well, later on when we go through the electron transport chain, FADH2 has enough potential energy to make 2 ATPs, while NADH has enough potential energy to give you 3 ATPs. However, it is not quite that exact, but we like to round it off to make it simple. Water comes in, and then the purpose of water coming in is to change the actual, you don't see it here, but there's a double bond that is actually going to break, which is going to facilitate malic acid to become oxidized in order for NADH to be reduced. And again, this NADH is also going to go to the electron transport chain. So here you're making 3 NADH per turn and 1 FADH2, and those are all going to the electron transport chain. But remember that you really are going to get 2 of these molecules coming in. So the actual net gain of NADH and FADH2 and ATPs in the Krebs cycle has to be really multiplied by 2, since there's 2 of them coming in. And here is what we just discussed as far as the net gain of your citric acid cycle. In cellular respiration, where you make the most ATPs is in the electron transport chain. And the way we make ATPs in the electron transport chain, again, is from the NADHs and FADH2 that you made during the glycolysis, intermediate steps, and the Krebs cycle itself. If you did the math, there's a total of How many? Well, let's think. You had 2 NADHs in glycolysis. You had 2 NADHs in intermediate steps. And you had 6 NADHs in the Krebs cycle. And you had 2 FADHs in the Krebs cycle. That will give you a total of 34 ATPs. And this is the way that you have these redox reactions occurring. NADH drops off the electrons, and through a series of steps of oxidation, reductions, ATP is made. In the electron transport chain, the ones that are doing the redox reactions are known as your flavoproteins, your ubiquinones, the metal container proteins, and your cytochromes. In the cell respiration, the final acceptor is your oxygen. In the anaerobic respiration, another molecule other than oxygen serves as a final electron acceptor. And here is your overview of how the electron transport chain occurs. As you can see, NADH becomes NAD+, and you have this hydrogen ion release. FADH2 becomes FAD, and he also has the hydrogen ions release. Now, as the electrons are passed through redox reactions from all these ubiquinone and cytochrome complexes, they go through redox reactions, and this energy is generated as is released, and it pumps these extra hydrogen ions right through the inner mitochondrial membranes. And as these hydrogen ions increase, it's just like you remember from diffusion. It goes from greater concentration to lower concentration right through this enzyme known as ATP synthase. And the word synthase comes from the synthesis, which is building together. And as the hydrogen ions come through here, ADP becomes ATP. Now you must say, okay, what about these hydrogen ions? Where did they come from? Well, originally they came from here. They were pumped across with the help of the electron transport chain of passing of those electrons, redox reactions. Now the final acceptor is oxygen. As you remember, oxygen is electronegative, so it has the ability to pull this like if it was a magnet, and then you make water, which is your final product that you remember from cellular respiration. Chemiosmosis is the process in which you just saw in a previous diagram. As those hydrogen ions are crossed, they're pumped across. That's the process known as chemiosmosis. And then, as they build in content, they go across the ATP synthesis to make ADP into ATP. Now, this is called oxidative phosphorylation. Very simple. NADH or FADH are being oxidized, and that process helps to make ADP become phosphorylated into ATP. The total yield that we get from the NADH and FADH is 34. And the reason behind that is because you have 10 NADH, and those give you 3 ATP each, so that's 30. But each FADH2, which is 2 of them, gives you 2 each, so that's 4. So that gives a total of about 34. There is other alternatives for organisms that don't have the ability to do the same processes. An example would be pentose phosphate pathway and the N-nodorodov pathway. The pentose phosphate pathway, as you can see, also begins with glucose. However, it goes through extra steps. And these extra steps are meaning more energy that you have to invest. So at the end of the yield, you will have less than you did with glycolysis. Sometimes cellular respiration cannot occur because oxygen is not present. As an alternate, fermentation occurs. However, some organisms cannot do cellular respiration because they are obligated, for example, anaerobic. Okay? When we have actually fermentation occurring, it does not happen in the mitochondria, it happens in the cytoplasm, in the case of eukaryotes, of course. What are the products that represent fermentation as far as organisms? You are most familiar with them. As a matter of fact, some of them are cheese, some of them are wine or beer or other types of alcohol, yogurt, soy sauce, and even rubbing alcohol or nail polish removers. One thing that we have to understand about this is that fermentation is not going to do the same yield as I mentioned to you earlier as far as ATP. Okay. For instance, in aerobic respiration, the potential molecules of ATP in prokaryotes is 38 versus 36 in eukaryotes. Okay, but fermentation is only 2, which are the ones that you actually make during glycolysis. Organisms do not only break down sugars, of course they do not. Some of them could be macromolecules such as lipids or proteins. And as far as protein and carbohydrates, they make about the same kilocalories per gram. But lipids make much more because they have many more hydrocarbons. And the more hydrocarbons that you have, such as the one you see in the fatty acid chains, the more actual ATP that you can release. For instance, from the fatty acid chains, they're all broken down into 2 carbon molecules known as your acetyl-CoAs, and each of those 2 carbon molecules go to the Krebs cycle after being processed through beta oxidation. In protein catabolism, the enzymes involved here are known as your proteases, and what your proteases do is that they actually break down the amino acids. Well, they break down the joint amino acids, and then they are deaminized because the amino end, it's actually going to be turned into urea, which will be released as waste. Now we're going into the anabolic pathways, and this is something that is the opposite of catabolic. Here we have photosynthesis, and here what photosynthesis does is first make the glucose for the glucose then to be broken down later on in order to make energy as ATP. Photosynthesis happens in organisms that have the organelle known as the chloroplast. Other anabolic pathways are the ones that require energy. Well, they all require energy rather, and the energy comes from other catabolic reactions that happened earlier. Here an example of the precursor metabolites and what they came from before we used them, or before they were used rather, in order to make the anabolic pathway occur. Here are the role of glucogenesis in the biosynthesis of complex carbohydrates. As you can see, it's sort of going backwards, right? Remember when we started with glucose before we in glycolysis, it was broken down through the catabolic systems to become into pyruvic acid, but now we're going backwards. As you can see, pyruvic acid is getting formed from the acetyl-CoA, from your acid fats, from your stored fat, that is, okay? And here the amino acids also come into play to form your G3Ps, and eventually you end up having glucose, and as your glucose are added together, monosaccharides become polysaccharides, and as you recall, those macromolecules will become your polymers such as starch or cellulose. Others from the glucose 6-phosphate will make glycogen, and from fructose 6-phosphate they will make the pectoglycan. which is the peptoglycan water you find in your gram-positive, gram-negative bacteria. In the biosynthesis of fat, which is your lipids, again, G3P is going to be formed. Acetyl comes in and releases what? The fatty acids. And as you remember, your glycerol and your fatty acids come together to give you your triglyceride, in this case fat. Synthesis of amino acids via animation, transamination. This is to indicate how your amination is adding the amino group, and transamination is to get that amino group together with another acid to form your different amino acids. Another example of an anabolic pathway is your nucleotides. Your nucleotides are the ones that make up the nucleic acids, and nucleic acids examples, as you recall, could be DNA or RNA. So when this occurs? Well, it all comes down to homeostasis. Whenever you need to make something, it's because you are lacking that ingredient, and therefore you get signals, intracellular signals, in order to make them. And whenever you need to break down something to make energy, you also get the signals to actually go ahead and break down that molecule to make energy. And that's what refers to metabolites. And this is a review of what you already learned. The use of allosteric sites with feedback inhibition. If you need something more, inhibition slows down. If you have enough, inhibition makes your actual anabolic pathway stop. Once you have enough of that product made, then it moves on to the opposite direction, which is catabolism. But each of these pathways are always involved with enzymes and coenzymes. The way these mechanisms are regulated is through gene expression. Gene expression refers of making of your protein. And this is from DNA sending the messenger RNA, transcribing it, and from the transcription it moves on to translation. And control metabolic expression is also through homeostasis and through feedback inhibition. Well, I gotta give you a big hint on that one. Why is feedback inhibition necessary for controlling anabolic pathways? Be ready to answer that. And this is the big picture of all of the metabolic pathways together. And as you can see, they all pretty much go to the backbone of cellular respiration. And glycolysis, of course. Microbial Nutrition and Growth, Chapter 6. Due to the microbial growth, it results in discrete colony or biofilm. A colony is one aggregation of cells arising from one single parent. Biofilm happens to be a collection of microbes living on a surface in a complex community. When we look at energy sources, we have to know what do we mean by photoautotrophs versus chemoautotrophs. Photoautotrophs means that they use light energy in order to make their own food, which is troph. Chemo means they use chemical sources. The photoheterotroph means they need the actual light to consume other sources from other organisms, and chemoheterotrophs, they use chemicals. such as aerobic respiration, anaerobic respiration, or fermentation, as we already learned. How do we determine the growth metabolism category? Do we fix carbon, or does it not fix carbon? Does it need light energy, or does it get energy from light? Does it get the energy for inorganic oxidation, or does it get energy from chemoautotrophs or other types of autotrophs or heterotrophs? What are your sources of energy? Sources of carbon. Remember that anything that contains carbon is known as organic. Organotrophs or heterotrophs, they get electrons from the same organic molecules that provide them the same carbon, or the same type of carbon rather. Lithotrophs are the autotrophs, which actually acquire electrons from inorganic molecules. Oxygen means they are aerobic, such as obligate aerobes. However, oxygen can also be deadly for obligate anaerobes. How can this be true? Well, toxic forms of oxygen are also highly reactive and excellent oxidant reagents. The resulting oxidation causes irreparable damage to cells. Four toxic forms. Peroxide anions. They are formed during the reaction of catalyzed Purine superoxide dismutase. They have hydroxyl radicals that result from ionized radiation and incomplete reductions of hydrogen peroxide. This is why we do in the lab the catalase test. When you see the bubbles, it means that they have the enzyme catalase and they can actually break down hydrogen peroxide, therefore releasing oxygen. You can see that happen, for example, when you have a wound. When you have a wound and you add hydrogen peroxide, you see the bubbles because you actually are able to break down H2O2 into water and oxygen. Oxygen requirements for aerobes, anaerobes, factory anaerobes, aerothermal anaerobes, and microbial. What are these? Well, if you were to grow these in loose media or broth, you will see how the organism displays growth or where they're highly populated. If they happen to be obligated aerobes, you will see most of the bacteria growing towards the top because they're seeking oxygen. If they have to be obligated anaerobes, they will grow towards the bottom of the tube in order to avoid the oxygen. A faculty anaerobe is an organism can do cellular respiration, therefore in the presence of oxygen, or fermentation, so it can grow throughout the tube. Aerothermal is somewhere in between. Now your microaerophiles have a different display. They will grow within the tube but towards the middle section of the tube throughout, not through the sides. Nitrogen is actually used very much for anabolism. Okay, if you don't have enough nitrogen, the anabolic reaction will cease. And that is because, as you recall, the amino end contains nitrogen in amino acids. There are other chemical requirements, and perhaps you remember, as you learned from the first lecture, that the basic elements for macromolecules of life happen to be carbon, hydrogen, nitrogen, and oxygen, but we also have phosphorus and sulfur. Trace elements are also present, and those are required only in small amounts. The reason they're required in small amounts is because if there are more than small amounts, they become toxics. The growth factors are the ones that are necessary for organic molecules that cannot be synthesized by certain organisms. such as certain amino acids that we get from or organisms get from the other sources. What are the physical requirements? Well, those are one of the requirements for the growth. Temperature. As you remember, when we looked back about enzymes, temperature has to be the one in which the proteins do not get denatured. Also, lipids have that requirement. For instance, if they're too low, the temperature that is, membranes become rigid and fragile. If the actual temperature becomes too high, the membranes of lipids become too fluid. And how does that affect? Well, the cell membrane has lipids, for instance. How did we determine that in science? If you look at the graph, the optimum temperature is determined in the growth that we do of bacterial in the incubators. And in this case, you can see that 37 degrees has to be slightly better than 30. But 22 degrees Celsius is too low. However, we have different types of bacteria. And some bacteria grow better at very low temperature, such as the psychrophiles. The mesophiles grow better at 37 degrees. Thermophiles and hyperthermophiles are the ones that can resist high temperature. The hyperthermophiles are very rare. Those are the ones that you find, for example, in hot springs. However, the ones that are more abundant and also they're more effective to humans are your mesophiles. pH, as you remember, its best growth in the case of in humans cells is around 7, and for the cells that get affected by bacteria is also around the same pH, 7. Above 7 is something that we call alkaline. Below 7 is acidic. Your neutrophiles grow best in the narrow range around the neutral pH also, just as we talked about in temperature, which is 37 degrees. The acidophiles, however, grow best in the acidic environment, such as in your stomach. And the granulophiles grow best in the basic environment, such as soils and water. Just as humans cells require water, microbes also require water, because water is used for hydrolysis. And as you recall, hydrolysis means hydro, water, lysis, to break down bonds. And when you break down bonds, you also release energy, or you break down macromolecules into smaller molecules known as monomers. The two effects of water are osmotic pressure and hydrostatic pressure. Osmosis is the diffusion of water, and your cells will carry in water or out of water, just like the same thing as in microbial cells, depending on the environment. And when we talk about the environment, you have to think about the outside of the cell wall or cell membrane. The best environment for the mesophiles is an isotonic environment, when the water flows in and out at the same rate and the osmotic pressure is obtained. However, if there's a whole hypotonic solution, you have actually a lot of solute inside the cell, so water is going to go in to dissolve it, and it makes the cell swell. If you have a hypertonic environment, you have a lot more solute on the outside of the cell membrane, so water will leave the cell and it makes the cell shrink. When we talk about physical effects of water, we're looking at the hydrostatic pressure. That is the water expels pressure in proportion to its depth. For example, the barophiles, they live under extreme pressure. The membrane enzymes depend on the pressure in order to maintain the three-dimensional functional shape. These are the type of microbial organisms that live at very deep depth. Biofilms. When we talk about biofilms, are a number of organisms, microbial organisms that live in association with different species. When we talk about association with different species, we talk about symbiotic environments. We have different types of symbiosis. The symbiosis could be when both are mutual and they both benefit, such as mutualism. That is synergic relationships. However, when one benefits and the other one does not, that is known as antagonistic, and those are parasitic. For instance, an example is how organisms get together and live in the same community, such as forming plaque on teeth. As you can see in the diagram, some microorganisms come together, and then as they form a matrix, then they start forming water channels in which the water flow. As you may recall, water is important as far as surviving the environments, not only for hydrolysis, but also for keeping the actual flow from one form to another. Inoculum. When we talk about inoculum, is when we actually get a bacteria from different sources. As you remember, ubiquity is when bacteria can grow everywhere. This could be from environmental specimens, clinical specimens, or even stored specimens. However, when we talk about culture, is when we're trying to cultivate organisms or organisms that actually are cultivated from one source or the other and transfer them from different plates or broth. From the first lecture, you remember that our bacteria grow from one cell and they duplicate, and the duplication process is known as binary fission. And every time they duplicate, they go through different generations. Therefore, it is algorithmic growth. When you first start growing a bacteria in a specific broth, we call it lag phase. Lag phase is when they start basically growing and get accustomed to the environment and to the actual nutrients that are in the broth. Once they start duplicating, they duplicate very fast. That is the log growth. That is the time that you actually want to transfer the organism from one source to another to keep them alive. Once they reach the stationary phase, they actually start growing and dying at the same rate. However, at that point, your bacteria also start generating a lot of toxins, and the toxins cause this bacteria to start declining, and that is called the death phase. And that will be the end of lecture 2. Have a good day.